Assalamualaikum warahmatullahi wabarakatuh. We are from Group 5 and we present our article review about genomics. For my article review, which is phenotypic and genomic characterization of Pseudomonas putida item 17297 spoiler of fresh vegetable, which is focused on biofilm and antibiotic resistant interaction of the species. So for that phenotypic characterization for the antibiotic resistance of the species, it is used. Two techniques were used, which is antibiotic susceptibility testing. It is triplicated by this diffusion technique on Mueller-Hinton agar 2 and manually sprayed on embryonic plates, and the discs were impregnated with antibiotics. These were placed on seeded embryonic plates within 15 minutes. Blank discs were used as negative control and the plates were incubated overnight at 30 degrees Celsius. The second technique is minimal inhibitory concentration assay. It is performed in sterile 96 well plates using MH broth added with increasing concentration of certain antibiotics. MH broth was poured in each well with inoculated medium in presence or absence of antibiotics. The assays were performed triplicate that added with an aqueous resazurin sodium salt in each well. The plates were incubated at 30 degrees Celsius for 24 hours. If a color change from purple to pink, it is indicated that they are positive bacterial growth. So for the biofilm, for the phenotypic characterization for the biofilm, also use two techniques, which is biofilm formation and metastasis assays. So the swarming and swimming experiments were performed in polystyrene petri dishes. The diameter of the swarming and swimming mortality zones were measured after incubation 30 degrees Celsius and 15 degrees Celsius for 24 hours, 48 hours, and 72 hours. The twitching mortality was assayed by subsurface agar method with slight modification, and the hazy zone of twitching mortality elaborated at the interstitial surface between the agar and the petri dish was visualized by staining with a solution of 0.1% wet per volume of crystal violet. The second technique is minimum biofilm eradication concentration assay. So the 24-hour biofilms form at both temperature, which is 4 degrees Celsius and 15 degrees Celsius in tubes. The concentration of each antibiotic equal of minimal inhibitory concentration valued of 2, 4, and 10-fold higher in MH broth were added to the test tube and incubated at 30 degrees Celsius for 24 hours. 6 replicates for each sample were carried out, and the MBAC was defined as the lowest concentration of antibiotics preventing regrowth of bacteria from surviving biofilm. The crystal violet staining was performed in conjunction with the MBAC assay to evaluate biofilm biomass by the remaining 3 replicates with 0.1% wet volume of crystal violet. So at the end of the research, the Pseudomonas putida item 17297 displayed resistant to several classes of antibiotics, and it also formed huge amount of biofilm. And for the biofilm lifestyle of Pseudomonas putida, as you can see here, it is more favored towards low temperature, and this indicates that the synthesis of enzymes related to spoilage will be promoted if the vegetables are stored in cold conditions. So the researcher of this research article hopes that more research will be done in order to identify more characterization of Pseudomonas putida towards the spoilage of cold storage vegetables. That's all from me. Next.
Assalamualaikum to Sir. My name is Dayang Kunar Atika Bintang Awang Ahmad. Today I would like to present my title, Functional Genomics Reveal Gene Regulatory Mechanism Underlying Schizophrenia Risk. Okay, for the introduction, genome-wide association study have identified over 180 loci that show strong association with schizophrenia. Most of the risk variants identified contribute to schizophrenia risk through affecting gene expression. Next, the main content that can be highlighted from this article consists of regulatory elements, which have a critical role in controlling gene expression. Second, about the functional genomic, which is used for identification of regulatory single nucleotide polymorphism. which is used to investigate the effect of schizophrenia risk variants on gene expression. Next is about the explanation carried out of pathway analysis of the target genes, and last about the uses of cell type specific expression analysis of the target genes. So, as a result from this article, figure A and B shows the number of the transcription factor disrupted by SNAP. From the finding, 132 risk single nucleotide polymorphism that disrupt transcription factor binding has been identified. About 97 were found associated with gene expression in human brain tissue. So, from these two pie chart and graph, a disruption of POLR2A, CTCF binding, and other transcription factor binding by regulatory SNAP have been identified. So, the discussion that can be explained based on the result, as this study identified 132 TF binding disrupting SNAP, this SNAP disrupt binding site of multiple TFSC, representing the most plausible causal SNAP. The regulatory effect of some identified functional SNAP was validated with reporter gene assay and allele-specific expression analysis. Besides that, nervous system development genes were significantly enriched among the target genes of the identified regulatory SNAP. Hence, it revealed new gene regulatory mechanism affected by schizophrenia risk SNAP, including widespread disruption of POLR2A and CTCF binding. This may be due to the possibility that CTCF and POLR2A have more binding sites on the genome than other TF. For the conclusion, it can be concluded that the investigation of the gene regulatory mechanism underpins schizophrenia risk was undergoes through integrating data from functional genomics. In this study, it provides new insight into the genetic mechanism of schizophrenia. Therefore, further mechanistic investigation and functional characterization of the identified causal variant and genes are crucial in improving the understanding of the pathogenesis and of schizophrenia. Okay, that's all from me. Thank you.
Assalamualaikum, sir. So my title is Cooperative Genomics of Protoploid Saccharomyces. Saccharomyces cerevisiae or S. cerevisiae is one of the yeast group. The study focused on S. cerevisiae in order to determine the differences between genomes of the species. So S. cerevisiae is the model system in this study, and the study had found a lot of S. cerevisiae species. The first achievement from the study is 5 species of Saccharomyces were found, and that 5 species we call protoploid. Protoploid is the division of Saccharomyces from S. cerevisiae lineage that happened earlier than its genome duplication. The 5 protoploid of S. cerevisiae share a core repertoire of approximately 3,300 protein families and a high degree of conserved synteny. The 5 protoploid yeast contain numerous copies of paralogous genes, either dispersed or in tandem arrays that altogether constitute a third of each genome. Next achievement, there are 3 species were found that have complete the genome sequences. The 3 complete genome sequences species are Cluveromyces thermotolerans, Saccharomyces cluveri, and Zygosaccharomyces roxy. And there are 2 species that have been compared its genome, which is Cluveromyces lactis and Ashbia gospi, and the comparison provide a multi-species of non-duplicated Saccharomyces. There are 2 methods, which is genome sequencing and genome annotation. The result is general sequencing strategy and brief description of the genome sequences, protein families and functional properties, and also orthologous genes and synteny conservation. Discussion: the genomes of the 5 species studied show common signatures that confirm Saccharomyces monophyletic origin and differentiate them from members of other Hemiascomyces subdivisions, which were used as outgroups for comparison. So The goal for this study is to characterize and compare gene content and genome organization of Saccharomyces that did not experience the ancestral whole genome duplication that sculpted the genome of S. cerevisiae and its closest relatives. So they determined the complete DNA sequence of these three novel yeast genomes in the table shown. Table 2 shows the summary of annotated features in yeast genome of interest. It's shown 8 species in the table, but we focus on the 5 protoploid species that represent 4 distinct clades of Saccharomyces, which is Zygosaccharomyces, Lankenkia, Clueberomyces, and Erythromycin. As they have complete phylogenetic relationship within Saccharomyces, they were designed as protoploid to distinguish from duplicated yeast. Next, table 3 shows the numerical distribution of protein families and corresponding numbers of protein coding genes, which they were able to determine the possible presence of segmented duplication of the genomes. In the table, proteins families were computed from 9 yeast species and were classified according to their presence in all species in column 1, subset of species in column 2, and only one species in column 3. To conclude from the study, the genome arrangement of Saccharomyces have provided more adjusted representation of various clades, enable the system to get a better understanding of its common properties and its individual varieties of the yeast. Saccharomyces were defined as have more balanced representation of different clades, providing a basis for understanding the origin of common properties of yeast community. So that's all for me. I pass it to you.
My name is Noatira Hanisah Binti Yaakob, and the title of the article that I will review is Genomic Correlates of Clinical Outcome in Advanced Prostate Cancer. Genomic testing can be performed on both biopsy tissue and tissue from the entire prostate. The various genomic and histologic characteristics of prostate cancer have been linked to a poor prognosis. We can look at the genetic makeup of the cancer may help predict whether your prostate cancer will grow slowly or aggressively. The genomic testing is performed on prostate cancerous tissue to provide information about how the prostate cancer may behave. This is the main content of the article. The first one is the genomic environment of metastatic castration-resistant prostate cancer has been well established. They present a systematic integrative study of 429 patients with MCRPC, genomic and transcriptomic profiles, histology, and clinical outcomes. The researcher identified groups of patients that may benefit from a more aggressive treatment plan, and the genomic and outcome results are made available to the research community for further analysis. There are many factors that make the genomic of prostate cancer have been hard to analyze in comparison to other cancer types. For results, it divided into two, which are clinical and histopathologic, and association of genomic alterations with clinical outcomes. For clinical and histopathologic, total of 429 patients were registered at one of 7 international concession centers. The whole exome sequencing was effective on 434 tumors, with some of them had underwent several biopsies. The RNA sequencing was successful on a subset of these 332 tumors from 323 patients. For the association of genomic alterations with clinical outcomes, the core feature of this dataset is concentrated the investigation on 18 of the most often altered genes and pathways. It also limited the clinical outcome study to patients who were taxane naive and starting treatment with a first-line ARSI for MCRPC. The bulk of genomic abnormalities in prostate cancer, however, do not yet have strong clinical applicability. Few experiments have linked particular genomic or molecular features to clinical results, but these have all been done in cell-free DNA or circulating tumor cells. Present an integrative study of genomic abnormalities, expression, and histologic evaluation in tumors from patients with MCRPC spanning the clinical spectrum of advanced disease with tissue obtained before and after treatment with ARSI and taxane. However, genomic testing has its own flaw, such as sampling issues, abstract data, and continuum of life. In conclusion, they provide an integrative study of genomic abnormalities, gene expression, histopathology, and clinical results in the largest single MCRPC dataset to date, and they make the data available to the scientific community for interrogation of genomic features in relation to outcomes. There are some widely available genetic studies that can be requested to offer more prognostic information for men with localized prostate cancer who are deciding on first or second line of treatment. Okay, that's all from me. Thank you. Next.
My name is Nur Azza Atira, and I will present the article review for the effect of genomic-related literacy on non-communicable diseases. Introduction. Genomic literacy can be defined as the working knowledge of genomic science in a society that includes an individual's decision-making, participant in civic and cultural affairs, and economic productivity. This study was conducted to find whether the genomic literacy can affect health status in individuals or not. Therefore, hypothesis was made based on the health literacy's effect on health status as it closely relates to each other. Main content. The association genomic literacy and health status was made based on multiple studies that have shown adult literacy skills with the ability to self-care, health service utilization, and etc. Genomic literacy should be expanded not just to health workers but to society as well. Okay, as we can see here, there are 4 levels of genetic contribution, which are genetic obesity, strong disposition, slight disposition, and genetically resistant contribution. Genetic obesity, the individual can be obese despite the environment, whether it is obesogenic or non-obesogenic. Whereas strong disposition, obese in obesogenic environment but overweight in non-obesogenic environment. Slight disposition is overweight in obesogenic environment and normal in non-obesogenic environment. Lastly, genetically resistant, which the individual will possess normal BMI despite the environment. Okay, based on these levels of genetic contribution, obesity, the individuals have the chance to change their environment to suit their genotype. Okay, Genomic Literacy Score (GLS) was used to measure participant knowledge on genomics. All participants had mean GLS of 14.2, significant difference of 7.2. Participants were grouped into their respective demography, which are marital status, occupation, and household income, which all three has significant difference for mean GLS, and parenthood status, age, and living condition, which has no significant difference for mean GLS. Participants who approve of genomic research has lower risk of hypertension and elevated GLS, whereas lower GLS group has higher rates of obesity and hypertension. No significant difference for diabetes and dyslipidemia, stroke, and heart disease. Discussion. The continuous unhealthy habits like overconsumption of salt, sugar, and energy may have led to rise in body mass index and blood pressure. However, there are possibility for reverse causality which may occur when the development of disease in individuals may spark their interest in learning genomic-related knowledge. Limitation in this study includes selection bias, higher prevalence of hypertension and obesity infection in the population. Conclusion. As a conclusion, this study has successfully shown that genomic literacy could be one of the risk factors for hypertension and obesity. A follow-up study must be conducted in order to obtain an accurate interpretation. And ultimately, genomic literacy can be used to assess risk of hypertension and obesity in medical care. That's all from us. Thank you.